Okay, here's part two. If you turn over to page 388, you'll notice that there's a section on another type of nerve cell called neuroglia. So we know about the neuron, but what we haven't discussed is the neuroglia. And neuroglia are these accessory cells in the nervous system, but they do not transmit an action potential. But what they do is they are like helper cells to the neurons. Now, there are four different types found in the central nervous system. That's this image. There's two different types found in the peripheral nervous system. I'm going to skip all that. You need to know it, but it's easy, so you can learn it on your own. The one type of neuroglia cell I want to talk about is the Schwann cell. And the Schwann cell is found in this picture on page 389. And you'll notice that these cells, what they do is they wrap around the axon of the neuron, okay? And what they do is they are like an insulator. So if you ever, you have an electrical cord, it has like a black plastic around it. That's an insulator for the wire that's inside, okay? That's actually transmitting the electricity. It's the same thing with the axon. Underneath this Schwann cell, which is the insulator, you have your axon, and that's where the action potential is traveling along. So what this does is it forms this sheath along the axon, and that sheath is called the myelin sheath, and it speeds up the rate of conduction. Okay. Now, you can have myelination in the central nervous system. That's carried out by a different cell you can read about. But in the peripheral nervous system, this is formed by the Schwann cells. Now, if you look at the image on 391, what you'll see, all this purple, this is the myelin sheath, okay? And see how it's wrapped layer upon layer upon layer upon layer. And it actually makes the action potential go faster because what it does is the action potential, instead of traveling along the axon, it jumps from node to node, because you'll notice there are these little nodes in between each Schwann cell. So it's like the difference between walking across a room and skipping across a room. So if you skip, it's going to go faster, just like it does here. That's called saltatory conduction. So when you get to the question about what's the difference between continuous and saltatory conduction, you know that saltatory conduction happens when you have a myelin sheath. Now, the outside of this myelin sheath, this very outer layer, is called the neuralemma. And the neuralemma is found only in the peripheral nervous system. And the neuralemma is responsible for repair of the neuron if it's damaged. Now, this does not happen in the central nervous system. And I've turned the page because this next page is all about regeneration of nervous tissue. And it tells you that in the central nervous system, you almost never have repair. Why is that? Because they don't have Schwann cells. And even though they're myelinated with a different cell. So you don't get this outer neurolemma that's going to cause repair and regeneration to occur. So it's sort of like if you have surgery and they cut some peripheral nerves and you don't have feeling for a few weeks, but then it comes back, you had regeneration of those sensory neurons, okay? But it's only going to be in the peripheral nervous system. If you have brain damage or damage to the spinal cord, that's permanent. Over on page 392, it goes through these 5 steps that talk about how regeneration occurs. That is on the test, but they've laid it out very nicely for you. I'm going to let you go through that. If I were you, I'd just read the little summary for each picture, know the steps in order of how repair happens only in the peripheral nervous system. The other thing I want to say about myelination, because there's a lot of lipids in this myelin sheath, it gives a white appearance to the nervous tissue. So when you hear that term white matter, what it means is it's referring to myelinated axons. When you hear the term gray matter, it means that it's referring to dendrites and cell bodies that do not have a myelin sheath. So now you know the difference between white matter and gray matter. Okay, right before we left for spring break, which seems like forever ago, we were talking about the neuromuscular junction, and we talked about how the neuron went to the muscle fiber. All these steps and things happened at the muscle fiber to depolarize it to cause the muscle to contract. Well, the same principles hold true for how an action potential, when I say that, I mean a nerve impulse, how that is going to happen on an axon. So I want to go through that very quickly. So if I've written this out on my whiteboard. So if you remember, now I had drawn these as muscle fibers, but now this is actually an axon. So if what we have is the axon right here, right? And here's our membrane. And along the outside of the membrane, we have a positive charge. And along the inside of the membrane, we have a negative charge. And we called that resting membrane potential. And in the muscle fiber, it was around minus 90 millivolts. And remember, voltage is just this separation of charges. That's all voltage is. So we've got a separation of charges, positive here, negative here. And the other thing we need to remember as far as ions go is there are more sodium ions on the outside of the axon, and there's more potassium on the inside. Remember, this is going to be important later when we have to move into depolarization. So the thing about resting membrane potential of the axon at negative 70 millivolts is that you have to have a stimulus come in to actually cause depolarization to occur. And what happens is you can have some type of graded potential perhaps come in. Now, your book calls these local potentials, and I sent a note out about that, but the local potentials are also graded potentials, and it's a small change. It's a small change that will not cause an action potential to occur, okay? Will not cause an action potential to occur. And what you need are multiple graded potentials to come into play so that you can actually hit what's called threshold. Now, threshold is when you depolarize the axon just a little bit so that it can hit this number that we're looking for, which is minus 55 millivolts. Okay, so we want to hit minus 55 millivolts to get to threshold. And what threshold means is if you can depolarize this membrane by the opening of some sodium channels, if you can get it to negative 55, then you can have an action potential. And the thing about an action potential is that it is all or none, okay? Now, a graded or local potential is not all or none. It's graded. That means it can be small, it can be large, it's graded. But action potentials, once you hit threshold, no matter how strong the stimulus or how weak the stimulus, if you hit threshold, you're getting an action potential. And the action potential is the same no matter what. And that's why we call it the all or none principle. Okay? So how is that going to happen? Well, once threshold is reached, then voltage-gated sodium channels are going to open. Now, remember, the sodium could not pass through the membrane. It had to go through a protein channel. And these channels open because the voltage changed. That's why they're called voltage-gated sodium channels. The voltage changed because of a stimulus or a graded potential, and when the voltage-gated sodium channels open, the sodium rushes inside the axon, okay? So you come down to my second image, and we have that region of depolarization. So when the sodium rushed in, what happened to those charges? They flipped. Okay, so now we have negative along the outside, positive along the inside. If you were to stick a voltmeter into that axon, what would it read? Plus 30 millivolts, okay? That's depolarized. And this region of depolarization is going to move along the axon, and that is your nerve impulse, okay? And we remember that in order to get the potassium out, okay, it's got to go through a voltage-gated sodium channel, and we have this depolarization occur. That's what's going to happen. The potassium is going to rush to the outside, okay? When we come back down here, we've got to repolarize everything, okay? So that region of depolarization is moving down. It's got to flip back, right? And so it's going to go through these gated channels. Okay, this is all described over here. See, here are your gated channels. There's this potassium gated channel 397. Okay, but this is all summarized in this chart on page 398. Okay, so you've got a local or graded potential, depolarizes it in the trigger zone to threshold. So what's threshold? Minus 55 millivolts. If it can get to minus 55, then these voltage gated sodium channels that are now in a resting state are going to open, and look what happens. All this sodium rushes in. Okay, and depolarization is going to occur. And then repolarization, the sodium channels inactivate, the voltage gated potassium channels open, sodium rushes out so we can repolarize it, and then they're all going to close. Now, if you can look at this, I think I drew the potassium gated channels in the wrong place, but actually they open for repolarization. But if you're looking at these, okay, so here is your membrane potential on the left. So here we are at resting state, minus 70. You hit threshold, there goes your all or none. There's your action potential all the way to plus 30. Okay, then we're going to repolarize, right? So here we coming back down. But you'll notice something happens right here. It actually goes down below minus 70. It may go to like minus 80. That's called hyperpolarization. It's hyperpolarized, and that's because the the potassium channels haven't returned to their resting state, and so it may be as negative as minus 90 millivolts. And if that's the case, if it's hyperpolarized, you can't really have another action potential come along. Okay. And this is exhibited in figure 1114 on 399. So it tells you what gates are opening, what gates are closing. It shows you the hyperpolarization, and it's telling you that you're now in what's called a refractory period. And a refractory period means that you can't have another action potential come along no matter how strong your stimulus might be. So there's two sections to this, though. There's what's called the absolute refractory period, which means no matter how strong a stimulus is, you are not going to get an action potential. But then there's what's called a relative refractory period. And what happens there is if you actually have a very strong stimulus, you can get another action potential during this period. That's called relative refractory. But you will not get one during the absolute refractory period. Okay, so you can read, this is all numbered, it's all labeled, it's a really good overview of what is going on. Over on page 400, it talks about the speed. We know that if it's myelinated and it has saltatory conduction, it's going to be a lot faster than if it's continuous conduction, which is much slower and non-myelinated. Okay, the other thing that they go over is what is happening at the synapse. Now this should look familiar because it should look like a neuromuscular junction. This isn't necessarily a muscle though. This is postsynaptic neuron. Well, that just means that here's a neuron and it's synapsing with another neuron. But you will notice that the steps are all the same as far as what happens at the neuromuscular junction. Okay, so you had to have an action potential come in. It opened these calcium channels. The calcium channels made the neurotransmitters be released. They were binding to the receptors in the postsynaptic neuron. That opened up your sodium channels and caused an action potential to occur. So all that's a review really from the neuromuscular junction, even though this may not be a muscle, the same thing happens at a synapse, no matter what the synapse is. Now, we always talked about, we're trying to get to threshold to have another action potential occur. But sometimes these neurotransmitters can actually be inhibitory, which means they don't want another action potential to occur. So we have these things called excitatory postsynaptic potentials, which would cause depolarization to occur and you have an action potential. But we also have inhibitory postsynaptic potentials, which actually make the membrane hyperpolarized so that you don't get an action potential. And so there's two options there. Your neurotransmitter may be inhibitory or it may be excitatory. So it just depends. You're going to have to learn about all the different types of neurotransmitters over here on page 411. And they just go through the different types. We know about acetylcholine because that's the one we saw at the neuromuscular junction. But there's a whole chart, and they tell you if they're excitatory or inhibitory, or they may even be both. So you can read through those, and you can learn what you need to know as far as the study guide asks you to. Okay, I'm at 14 minutes, so I better stop. But I'm at the end of 11. So if this was helpful, let me know. And if it wasn't, let me know. But I can do it again on any other topics if need be. good luck, study. Please don't wait till the last minute. It's very important that you do well on these last two tests. I hope everybody is safe and well. Don't go anywhere, and good luck.